All right. Hello. Let's talk protein. So, protein sequences. We have a lot of them. We're going to use a lot of them in the class. Where do they come from? That's a great question. Um, there are a couple different ways you can actually sequence a protein, but for the most part, they are not actually sequenced. We infer them. So, the direct sequencing method is called Edman degradation. You can actually put in this chemical and it will react with the end of the protein, so whatever's at the end terminus of the protein, and it will break off the amino acids one at a time. And so, congratulations, you've broken off some amino acids, but then the follow up is that you do mass spectrometry, which is basically a way that you can measure the mass of different molecules. And so you'll throw this thing basically at the wall. And it's like if you had a ping pong ball, so a little hollow ball, and let's say a tennis ball, and let's say a baseball that's sort of hard and heavy. If you throw these things just with your arm, which one's going to go fastest? The ping pong ball is going to go kind of slow because it has very little mass and it may not even make it to the wall. The tennis ball, you can probably get to the wall, but it probably won't be as fast because it's kind of light. And then, if you had a nice heavy ball or a rock shaped like a ball, you could probably really fire that thing in there and it would hit hard. And so this is how it works. You accelerate the proteins, and depending on, or the peptides, the little amino acids, depending on how much mass they have, they fly at a certain rate, and you measure how long it takes for them to hit the detector, basically. And from that, you can calculate the mass of the thing that you threw. Yeah, it's sort of indirect, but it's very useful, and it's good. I like it. So yeah, this is the way you can sequence a protein, but you can only sequence the couple of amino acids at the end. You can get maybe 5, 6, 7 amino acids. That's about it. So if you already have a very good idea which protein you're dealing with, that can give you the last piece of information. You can say, okay, I've got it now. But there's also a better way to do this. And so this involves some inference. It's not as direct. But you can take a whole sample of protein. You can even take a mix of many different proteins. You can separate them, and it's a process called liquid chromatography and then two-dimensional mass spec. So they'll call it LC-MS for liquid chromatography, mass spectrometry again. How it works is you're going to take your sample, you're going to separate it out, separate out the proteins by size, run them on something like a gel. Then you're going to take chunks out of the gel that probably contain multiple proteins but of similar size, or at least similar electrophoretic mobility, which is like how fast things travel down a gel. It's not always exactly the same as size. Sometimes charge plays a little role, and sometimes shape plays a little role. Then it's hard to actually get the structure or identification of an entire protein. So what they'll do is they'll break it into peptides. And so here's a trypsin digestion. So that's a protein cutting enzyme that'll cut most proteins somewhere. So that'll break up your big proteins into little proteins, which we call peptides. And then these little proteins are what's going to go for mass spectrometry. So the first thing that happens is we're going to do liquid chromatography, which is again going to separate out these proteins, so that proteins that are identical end up together and proteins that are different are going to end up in different places. So basically they're going to be coming out. We're going to be sending them out of a nozzle, but at a different time. So all of one peptide should come out together and should be sprayed out the nozzle. So they're going to get sprayed out in these little highly charged droplets, and they're going to go into the mass spectrometry detector. And you can look up the physics of it. It's even on Wikipedia. It's outside kind of the scope of this course. We just need to know that it exists and, that it is reliable, basically. So that'll give you the rough mass of the peptides. And so that'll be step 4. Here's our mass spectrum. So we got an average mass of 516.27 Daltons, which is great. Or rather M to Z, which can be converted into actual mass. And then the second dimension is we're going to take those proteins that have been separated by mass spec, and we do mass spec again. So you send them again flying through the air, but in a way that's really harsh, and it's going to break up the proteins. And it's going to break them from the C-terminal end for the most part. So you're going to chop off 1 or 2 or 3 or 4 or 5 amino acids from the C-terminal end, and you're going to send all these fragments out. And so you know the total mass of the fragment from step 4, and then you know a little bit about which amino acids are in the fragment from step 5. And with that, you actually get a partial sequence. You see the sequence comes out kind of backwards here, but yeah, that's like addition of each additional amino acid. Push it up here. So there you go. And so this is a technique that is widely used to estimate which proteins are present in a sample. Unfortunately, we can't really be any more direct than this. You can look for individual proteins, but each of those tests takes a while and a fair bit of money, and so people generally don't do that. They'll take the results from this with a grain of salt as being pretty good. Yeah. All right. But how do we actually do this? Because those are both ways to see what protein is in a particular sample if you already have some idea of which proteins should be in that sample. But what if you want to compare all proteins? Well, the answer is you infer that. Yeah. So since we know that there is a genetic code, and we have to be careful because the genetic codes of, for example, mitochondria and some bacteria are mostly the same but a little bit different. They'll have some codons that code for different things than we would normally get here. But yeah, we just we figure out what could be translated from a given stretch of DNA. And then we're going to use that logic. We're going to say, okay, what's a long thing? Have we got something that's statistically should not exist because it's way too big to be random? That's a good idea that it might be a gene. Have we got something that's conserved that we find in other organisms, or that somebody's actually done an experiment and proven actually exists? In which case, if you have something that looks similar to that, you can say, you know what, probably that's okay. So once you have that sequence, one of the best ways to see whether your sequence is similar to something else, because if you pass the phylogeny criterion, the rest doesn't really matter. That's generally good enough. There's something important about this if it is conserved. So you can use your search engines like all the different versions of BLAST, or HHBlitz is actually a fantastic search engine. The BLAST programs will find things that are fairly close to what you're putting in, and HHBlitz will find things that are actually fairly different and fairly distant matches, but it will still find them. So it's like the big guns you can bring out. All right. And then if you already know what gene, if you know it's say a pyruvate kinase, for example, which is what this little spinning drawing up here is, then you can say, okay, I'll get a whole bunch of pyruvate kinases and you can align them. And so these are some of the programs that do aligning. Of these, they're all available online except for Mega. You would have to install that on your computer. So let's not worry about Mega. And there's a bunch of other programs as well that you can install on your computer. But yeah, free online stuff. Clustal Omega does a good job. So there you go. All right. So what are these things actually doing? Yeah, well, here you go. So there are several tools that will translate protein for us and help us figure out which parts of the protein are probably actually translated to keep the organism alive, and which things maybe could be translated but probably aren't important. So the two main ones that I would use for this would be ORF Finder, that's ORF Finder, and I also like XBasy Translate because you can play around with the settings, and it's very nice for sort of detailed work where you have to fix something, for example. All right, so any given piece of DNA has neither a front nor a back. It just has two ends. And since it's double-stranded, remember the two strands in double-stranded DNA or RNA are pointed in opposite directions, like that. So you see you got a 5' running toward a 3', 5 front, 3 tail is how you can remember it. So that's the front of this part and the tail of this part, but that's the front of this strand and the tail of this strand. And we can't tell whether we've got this thing right side up or basically upside down, whether this is the meaningful strand or the bottom one is. So we're going to translate it in all 6 reading frames. And I'll get to what that means in a second, but that's what they're showing up here. All right. So there's no marked starting place for DNA. Like you can't just look and say, oh, AGGCT, that's got to be the front end. Yeah, that does not work. There will be an ATG somewhere. And if there's an ATG, like here's one in the middle, ATG, that is a potential start place, but it also could be the reverse complement. It could be somewhere in the middle because proteins have a methionine, which is what you get from ATG in the middle quite often. It doesn't necessarily mean anything. So yeah, there are 6 ways of reading the protein. We call those reading frames. And what we're looking for is a long open reading frame. So open means there's no stop codon. And so you can see that all the stop codons are marked with little stars here. So like TAA is a stop codon. There's TAA. And so yeah, that's how it works. An open reading frame would not have a stop codon. So looking at these 5 open reading frames, there's 3 upside down. Look, that one has a stop codon, that one has 2 stop codons, this one has a stop codon at either end. So those are all not open reading frames. And then the first one here, stop codon, and the third one, stop, stop, and stop. But look, the second one in this direction, no stop codons. So it's not definitive, but this might be something interesting. So this is like the first step. Let's see if there's something interesting. So we can name these different reading frames, and I just want to make sure you've got a good picture of what I mean by reading frame. So we read DNA, which gets turned into RNA. So really we're reading the RNA, but we'll just pretend that the RNA is a direct copy of the DNA with no funny business. So we'll read it in groups of 3, because remember, every 3 nucleotides gives us 1 amino acid of a protein. And those are called codons. But we can't tell which one we should start on. Pardon me. So we could start on A, and we go ATT, and then TAC, and then AGG, and we could call that frame 1, 5' to 3'. Or we could say, well, what if it doesn't start on the first A? What if it starts on the first T? So then it would be TTT, then ACA, then GGG, and so on down the thing. So okay, we call that frame 2. And what if we said it starts on number 3? So that's TTA, CAG, GGG, and that would be another frame. Each of these frames would translate a completely different protein that basically has nothing in common. So there's the 3 forward and then the 3 reverse. So starting AGG or starting GGC or starting GCT. Now, why do I say there are only 6? Because you could say, what if I start on the 4th one? Well, if you start on the 4th one, that's the same as starting on the first one, but leaving out the first codon. And so don't leave out the first codon. So there's 6 reading frames that actually apply, and all the rest are subsets of those 6, if that makes sense. I hope it does. Yeah. All right. So that's how reading frames work. And so in the next section, we'll go into a little bit about proteins in general and how we can use some of this logic to figure out what they're doing.